So, information presented is not an official opinion of Michigan State University or the University of New England. I'm Zach Burton. My co-host is Lele. That's Lele. And we're completely responsible for content. Yep, there it goes. Okay, so this is a Life on Earth, Origin of Life webcast. And the title is Zombie Models Shamble On. Lele is a professor at University of New England, Biddeford, Maine. He's in the School of Biological Sciences. I was a professor for 30 years at Michigan State University, now retired, in the Department of Biochemistry and Molecular Biology. So the subliminal messages here. Looks like some of them have gotten out of my screen. Anyway, I'll go bottom up. That way, maybe I'll get them all. The first one is that I don't like codon-centric genetic code models. I like anti-codon-centric genetic code models. This is a messenger RNA first model. tRNA first models are better. Two mini helix models for evolution of tRNA. Those are falsified models, and I'll try to make that point as strenuously as I can. I would like to kill the Urzyme hypothesis. This has been around for about 10 years, but it's not a good model for various reasons. Accretion models for tRNA evolution, those are not good models. And the reason accretion models are not good is that they're chaotic models and tRNA evolved by a completely ordered process. Any model that involves bringing in an intron early and depends on intron sequence can't be right. So introns and tRNAs can be ancient, but in any model for tRNA evolution that involves an intron sequence can't be correct. Up there, out of my sight, is convergent models. Convergent models are not allowed, and I'll try to explain why that's so. Okay, so this is the greatest story in about 4 billion years. It's really fascinating. It's fascinating to us, and I hope it's fascinating to a wider audience. We're presenting a puzzle that just about anyone can solve, and the puzzle is solved here. The 331 nucleotide mini-helix theorem explains tRNA evolution. And it explains it for all time. So this is a theorem, not just a hypothesis. It's a proven hypothesis. We're relating recorded history from about 4 billion years ago, and it's recorded in genetic code. So the reason it's recorded is the primordial sequence of tRNA, type 1 and type 2 tRNA, is known with certainty. And this theorem describes that evolution. So this involves identification of root sequences, and these are root sequences that go back to pre-life. So these are really ancient sequences that were recorded in genetic code and never overwritten. So lots of people don't like biology, but they like language and linguistics and logic and various other things, or they like artificial intelligence. This is really a story about evolutionary intelligence or biological intelligence. So evolution learns to solve problems. So it involves learning. And this is an example of evolutionary learning. very similar to artificial intelligence, but because this is biological, it's not really artificial. Okay, so these are the models that I wish to kill. So zombie models. The Erzheim hypothesis is not correct, and as we go, I'll try to explain more about what this is about. But class 1 and class 2 aminoacyl tRNA synthetases are simple homologs, and that contradicts the Erzheim hypothesis, but this is absolutely certain. And I'll show you that a couple of different ways. An accretion model is one where, for instance, tRNA would grow a stem in a loop at a time, like a little plant. Such models are not allowed, and I'll try to explain why for tRNA early evolution. And one reason is that primordial tRNA sequence is completely ordered, 100% RNA repeats and RNA inverted repeats. So there's this Ouroboros theory. Di Giulio has a model. Di Giulio's model has several things wrong with it. It's a 2 mini-helix model. It's a convergent evolution model. The Ouroboros model is a convergent evolution model, and none of those can possibly describe the evolution of primordial tRNA. All 2 mini-helix models are out. Okay, so Di Giulio is a 2 mini-helix model. There are other 2 mini-helix models. They can't possibly be right. And one reason they can't be right is that tRNA evolution requires a 3 mini-helix model. These models are incompatible with the anticodon loop being a homolog of the T loop, but very clearly these two sequences are homologous. And in addition to this one and that one, that adds up to 2. 1 plus 1 equals 2. You have to consider the D loop, and so that's 1 plus 1 plus 1, and that equals 3. You require a 3 mini-helix model. So they're mRNA-centric genetic code models, codon-centric genetic code models. These models are not as good as tRNA-centric models. These are much better. So if you read this literature, you'll find many of these genetic code models with a complexity of 64 assignments. tRNA-centric model has a complexity of 32 assignments. And so at the base of the code, this view, an anti-codon-centered view instead of a codon-centered view, is a much better view of evolution of the genetic code. It's not that these are wrong necessarily, but they're too complicated and they're too degenerate. Any kind of intron first model that relies on intron sequence can't be right. So at least one of Di Giulio's models is based on an intron sequence. That can't be right. And it can't be right because of the sequence of primordial tRNA. So including an intron in the sequence is not helpful for figuring out how tRNA evolved. Any kind of convergent model. So I should explain what a convergent model is. So if you take, for instance, 2 little pre-tRNAs and you expand them both, and then both of them expand to the same form, that would be a convergent model. That's not a reasonable model. Basically, you have to start with the whole molecule, or type 1 and type 2 tRNAs, and then diverge to the complete tRNAome. That's the only kind of model that makes sense. So lots of models are convergent models. The Ouroboros model is a convergent model that falsifies this model. Di Giulio's 2 mini-helix model is a convergent model. It can't possibly be right. And there are other convergent models as well. Some people put forth convergent models without even knowing that that's what they're doing. Okay, so we're going to kill some zombies. And the first approach will be to show that class 2 and class 1 aminoacyl tRNA synthetase enzymes are homologs, simple homologs. That means that these are genetically related to each other in a straightforward way. And so because this is true right here, this is false. And I'll try to explain as I go. So this is a class 1, and this is a class 2 aminoacyl tRNA synthetase, GlyRS and ILURS. And these sequences show the genetic relationship of this enzyme to that enzyme, and it's a simple relationship. In order to find this relationship, you can't look at any old ILURS and GlyRS. You have to look at specific ones from ancient archaea. But ancient archaea are the oldest organisms and the most conserved from the last universal common cellular ancestor, LUCA. Okay, so in red, you have identities, I equals I, K equals K, G equals G. In yellow, you have similarities, so S is similar to T, Y is similar to F. And these alignments are very good alignments. They're fully in phase, no gaps, and they have an E-value statistical score of 4 times 10 to the minus 12. So if you take the reciprocal of that number, you get 2.5 times 10 to the 11th. So there's, just based on this evidence alone, which is not the only evidence, there's one chance in 2.5 times 10 to the 11th attempts that these two sequences are not true homologs, that this is an accidental alignment. So the heavy Las Vegas betting is on homology. It's not on the Erzheim hypothesis. This is playing the very long odds. I don't know if you go to Las Vegas and play the 1 to 2.5 times 10 to 11th odds, but I would recommend against it. It might have a good payout if you win, but you're not likely to win. Okay, so just to beat a dead horse, I'm going to explain that class 2 and class 1 aminoacyl tRNA synthetase enzymes are simple homologs. And here's more evidence of this. So before I showed you the alignment of two sequences, here I'm showing you the alignment of 1, 2, 3, 4 GlyRS from ancient archaea, and 2 VAL-RS and 3 ILU-RS. So these are all 1A enzymes. This is a 2A enzyme. Where you see red, that shows homologous alignment. So these 1A enzymes have an N-terminal extension. And then you pick up the homology, which is over most of the length of the GlyRS2A. Some of these enzymes have slightly different C-termini. Here you have a zinc finger that's in common between the different proteins. That's another demonstration of their homology. You have a zinc finger that's unique to class 1A enzymes, and that's located here. This is a fairly typical zinc finger. This is somewhat more unusual zinc finger. But basically, the fold of the 1A enzymes is different from the 2A enzymes because of the N-terminal extension and because of the environment of the zinc 2. And that changes the whole fold of the protein. In the case of GlyRS2A, the zinc finger here initiates the class 2 fold. Okay, so I've described this before, and I'll do it quickly here. The 331 nucleotide mini-helix theorem, and this is a theorem. It's a proven hypothesis. So there are no theorems in biology, but I suggest that this should be generally and universally accepted as one. For one reason, this describes evolution of life on our planet in a way that it's never been described before. So here's type 1 tRNA, and you can tell it's a type 1 tRNA. It's colored for the model that I'm describing. The variable loop here in yellow, that's 5 nucleotides, 1, 2, 3, 4, 5, and it connects up to this cyan segment here, which is a stem for this stem loop stem, the T loop. So these 5 nucleotides correspond to these 5 nucleotides genetically, and this is 5 out of 7 total. And here in green, that's 7 total. And these 5 here correspond to these 5 here in green. So this is based on a GCG repeat, GCG repeat, CGC repeat, CGC repeat. This is based on a UAGCC repeat, that's the D loop. Here you have a stem, loop, stem, stem, loop, stem. This is a homolog of that. This loop is 7. That's a U-turn after a U. This loop is 7. That's a U-turn after a U. And here you have the sequence at the 3' end, A, C, C, A. The 2 C's attach the tRNA to the ribosome in the A and P sites by forming Watson-Crick GC pairs. The discriminator base is A. That's the oldest discriminator base. And you can see this is a tight loop. It's a tight loop because you have a U-turn after a U. It's all snugged up. It presents a 3 nucleotide anticodon. So this is specialized biological intellectual property, if you want to think about it that way. This is a type 2 tRNA. And going back to a type 1, here you have a V loop that's 5 yellow nucleotides. And here you have a V loop that's 7 yellow nucleotides plus 7 green nucleotides. So these 7 here relate to those 7 there. These 7 here relate to these 7 here. These 5 here relate to those 5 there and those 5 there. So this is based on a GCG repeat, GCG repeat, GCG repeat, CGC repeat, CGC repeat, UAGCC repeat, stem, loop, stem, and a homologous stem, loop, stem. There you have a U-turn after a U in a 7-mer loop, 7-mer loop, U-turn after a U. This is a somewhat unwound loop because of the structure this was taken out of. It should be on the ribosome, it would be a tight loop like this one. And here you see these 5 nucleotides there that correspond to these 5 here. That would be these 5 here, 1, 2, 3, 4, 5. And you would delete these 9 and link this space up here to the cyan piece to make a 5 nucleotide V loop as in type 1 tRNA. Okay, so we have polymer world, going to mini helix world, going to tRNA world, and I've talked about this at length in other webcasts. Everything on this slide is absolutely certain. The only slight uncertainty is exactly what the pre-life sequence of these red loops are. This is actually a good guess. There's the U-turn after a U, probably a C in that position and A in that position. This position is probably an A, more uncertainty with these three nucleotides here. But at LUCA, at the last universal common cellular ancestor, which I'm going to say has a fairly intact genetic code, this sequence is CU, U-turn, B, that's a G, a C, or a U, not an A. There's never A in a wobble position. Two N's, that's A, G, C, or U, A, G, C, or U, and two A's. And this sequence here would be U, U-turn, C, A, U. This is especially selected to support this particular loop geometry, which is slightly different than the geometry of this loop. Okay, so to count the three mini helices, you can count them here, 1, 2, 3. You can count them by mini helix cores, 1, 2, 3. The anticodon loop is a homolog of the T loop. Anticodon loop is a homolog of the T loop. This is the D loop. So 1, 2, 3, 1, 2, 3, 1, 2, 3. There are many ways you can count up to 3, but it can't be 2. Okay, models that try to make it into 2 have to be accretion models, and those can't be correct. So is there an argument against this model? So the arguments against this model would be that this stem loop stem and that stem loop stem are not homologs. They're obviously homologs. Another argument would be that this GCG repeat is not a GCG repeat. This GCG repeat is not a GCG repeat. Clearly, that's not right. That this CGC repeat and this CGC repeat are not CGC repeats. And that this CGC and this GCG repeat are not CGC and GCG repeats. So the arguments against this model are ridiculous. People have actually put some of these into print. I'm not sure why they did that. Okay, so we're back to our zombie model. So the Urzyme hypothesis and the counter argument is that class 1 and class 2 aminoacyl tRNA synthetases are genetically related to each other as simple homologs, as I showed you. In the Urzyme hypothesis, basically class 1 and class 2 genes would have been opposite strands, and then those opposite strands, which made things called Urzymes, subsequently evolved to class 1 and class 2 aminoacyl tRNA synthetases. But what I told you is that they're simple homologs aligned in the same direction, 5' to 3', and I proved to you that that was so. So I'm hoping that this model will die. I keep getting papers to review based on this model, and I think that they should read more broadly and read some of the falsifications that we've offered for their model. Okay, an accretion model, you have, let's say, a stem loop stem for the anticodon loop, and then the rest of the tRNA grows around that. That's a chaotic model. It's a random sequence model, and no random sequence model can possibly be correct. The primordial tRNA sequence is completely ordered. and it requires that tRNA be formed, type 1 and type 2 tRNA be formed in a single attempt, couple of attempts, and then you diverge the entire tRNAome from those preformed tRNAs. So only the 331 nucleotide mini-helix model works. The Ouroboros theory, the Di Giulio model, any two mini-helix model, this is a two mini-helix model, can't be correct. This is a convergent evolution model, and this is a convergent evolution model, and those are not allowed. All two mini-helix models are falsified, so that's a two mini-helix model. There are others. For tRNA evolution, these are incompatible with the anticodon loop, the T-loop homology, and also with the presence of the D-loop. The D-loop is a third. So if you add the D-loop to the T-loop to the anticodon loop, you get three mini-helices. You require a three mini-helix model. Okay, so these models are not falsified, but they're also not good for the base, describing the base of the genetic code. So a codon-centric model is out, and an anti-codon-centric model is much better. So think of it this way: if you read a paper that's describing this, it's too complicated of a model, and by a factor of 2, and it'll be confusing. It'll be overly degenerate, if you want to think about it that way. No intron first model. This is at least one of Di Giulio's models is an intron first model. Can't be right. And it can't be right because of the primordial sequence of tRNA, which we've described. A convergent model, you start out with little sequences and you grow from those to big sequences, but then those big sequences have to have the same form because they're all tRNAs, and all tRNAs have the same form in ancient organisms. So basically you have to start with this and then copy it and diverge it to make a tRNAome. That's the way tRNAomes evolved from primordial tRNA sequences. Okay, so please allow zombie models to rest in peace. It's time that some of these died. And now I can see convergent models aren't allowed, intron first models aren't allowed, accretion models are not reasonable and aren't allowed. The Erzheim hypothesis I killed a couple of different ways. Two mini-helix models must die. Codon-centric genetic code models aren't as good as anti-codon-centric genetic code models. And with that, I think I'll stop and see if there are questions. Do you have questions, Lei?
Yes, I do have questions. So you described all these zombie models. I do have several questions for you. And can you reiterate what's the Erzheim hypothesis and what's wrong with it?
Okay, so what's the Erzheim hypothesis? This has been around for a long time, and many papers have been written on this hypothesis. But basically, the idea is that you have two strands of a nucleic acid. I don't know if that's RNA or DNA, and one encodes for class 1 and one encodes for class 2 aminoacyl tRNA synthetase. And then after you make these transcripts, these things evolve separately to their final forms. But what I showed you is that the sequences align like this in the same orientation, 5' to 3'. So they're simple homologs for class 1 and class 2 aminoacyl tRNA synthetases. Class 1 are twice as long approximately on the N-terminal end.
So my second question is, what is a convergent model for tRNA evolution?
So a convergent model would be like the Ouroboros theory, for instance. And in such a theory, you have these two small RNA rings, and they're independently going to grow to tRNAs, but the tRNAs they're going to grow to will have the same form and nearly the same sequence. So that's not a reasonable model. Any kind of model that involves convergent evolution. So DiGiulio's two mini helix model, the T loop and the anticodon loop have to converge on each other's form and yeah, in the tRNA, and that's not a reasonable model. You can't have two sequences converging in tRNA. And one reason is the entire primordial tRNA is ordered.
My next question has to do with the intron first model. Can you explain a little bit more? What is it? What is the intron first model for tRNA evolution?
Okay, so introns in tRNAs are ancient relics of jumping genes. So introns can be very ancient. They can be more ancient, for instance, than tRNA. But basically, the 331 nucleotide mini helix model fully describes type 1 and type 2 tRNA evolution, and there's no need for any kind of an intron sequence. So any kind of tRNA evolution model, like one of DiGiulio's models, that relies on intron sequences can't possibly be correct.
So how can you be sure that two mini helix model are inadequate for tRNA evolution?
Okay, so two mini helix models. So the problem with two mini helix models is this stem loop stem and that stem loop stem are obvious homologs.
Mhm.
And so that's one and that's two, but there's also this one, so that's three. So a two mini helix model can't reasonably be correct. There are two different flavors, so this flavor and that flavor, these are initially identical sequences, what becomes the anticodon loop and the T loop, but this one's a different flavor with a different sequence, different core sequence, same flanking sequences.
Yeah. So can you explain more what's wrong with the codon-centric model for genetic coevolution? And why are anticodon-centric models better?
Okay, so I can try. Okay, so what's wrong with these models? So here the complexity is 64, here the complexity is 32. So this is half as big a problem as this problem here. So that's an advantage of tRNA-centered model or an anticodon-centered model. I'm going to say that the anticodon comes first in evolution of the genetic code, and the codon is secondarily linked to tRNA specifications of anticodons. So in a way, maybe these should be called codons and these should be called anticodons, but it's too late to change that now. Okay, so the anticodon comes first, the codon comes second in evolution, earliest evolution.
I think those are the questions I have now.
Okay, well, let's then stop the share and stop the recording. Thanks everybody for your attention.